Hi everyone, I hope you are doing well. In continuation to our previous discussion about the T cell activation, differentiation, and memory, today we are going to discuss about the T cell superantigens. Very interesting molecules, and they play a big role in providing or in creating some diseases or some disorders in the body, right? So let's start these super molecules known as the superantigens. So when we talk about the T cell superantigens, they are special T cell activators or unconventional antigens. So when we say unconventional, that means they are not the conventional type of antigens, all right? And they are the microbial proteins. They are the proteins from the viruses and bacteria. Okay, so they are the activators of the T cells, and they stimulate a primary T cell response which is similar in magnitude to a response to allogenic MHC molecules. So just like the MHC molecules which are allogenic, they can give the kind of response, they can give the kind of stimulation to the T cell. These antigens, they can also do the same thing. They can also stimulate a primary T cell response of the similar magnitude. Okay, and they recognize the immune receptors outside their usual recognition sites, for example CDRs. So if you see, and then we are going to see through the picture that their recognition sites are outside the usual recognition site. Okay, now T cell super antigens, they cross link the TCRs and MHC class 2 molecules on the antigen presenting cells. So antigen presenting cells, they carry the peptide molecule. Okay, and they usually display with class 2 molecules here. Okay, while the TCRs, they are present on the T cells, and when these T cells and MHC class 2 molecules, they come together, then these T super antigens, these T cell super antigens, they can cross link both. All right, so they not only bind to the TCRs but also bind to the MHC class 2 molecules, not class 1 molecules, remember this. So by binding, they can recruit the lymphocytes. Okay, they can induce cytokine storms. Cytokine storms is a phenomenon where cytokines are released or they are secreted in a very high concentration. Okay, and that can give or that can induce a very difficult situation inside the body. They can also cause T cell energy or apoptosis. So, energy is what? It is the unresponsiveness of the T cell. All right. And apoptosis is the programmed cell death. So, T cell superantigens cross linking can cause these kind of incidents inside our body. Now, these superantigens, they have the ability to hyperstimulate T cells, right? How? You see the difference here. If you see the normal adaptive immune response, then the percentage of the activated T cells, it is somewhere around 0.0001% in case of the normal adaptive immune response. But when the exposure is superantigen, okay, when superantigen stimulates, then the immune response, it causes up to 30% of the T cell activation. So can you see, can you imagine here? Here the normal is just 0.0001% of the cells are stimulated. But superantigens can stimulate up to 30% of the total T cells. So how much harm they can make, they can do inside the body. Now, because they bind simultaneously to the specific variable beta regions of the T cell receptors and to the alpha chain of the MHC class 2 molecules, which you can see in the picture here. This is the T helper cell, and T helper cell, it has MHC class 2 molecule. So you can see, oh, sorry, it has, the T cell receptor, and antigen presenting cells, they have the class 2 MHC molecules, and these class 2 MHC molecules, they always bind to the TCR at the T helper cells. So you can see here, this is the T T cell receptor alpha chain, beta chain, and this is the MHC alpha chain, beta chain. So this super antigen, it is binding the variable beta chain fragment of the TCR and simultaneously to the alpha chain of the MHC class 2 molecule. And as I told you that they don't bind to class 1 molecules here. So once they bind, they do a clamp like connection. Their connection is like, clamp like, and that mimics a very strong TCR MHC interaction. So actually they are binding outside, the zone, but they clamp in such a way that they mimic a very strong TCR MHC interaction. So this interaction between, the TC, the T helper cell and APC, and then it is fortified by the super antigen that induces the activation without the need for TCR antigen specificity. They don't need that. Okay, and because of this, because it is non-specific, the activation is polyclonal. Here, if this would have been a very specific interaction, then this would be a monoclonal activation. But super antigens, because they don't need very much this TCR antigen specificity, and they can induce the T cells, therefore the activation is a polyclonal activation. So any T cell which is expressing a particular V beta sequence, that will be activated. All right, now there is no need for specificity. They just need this specific V beta sequence, any particular V beta sequence, and that super antigen can bind and then it can activate. So that leads to a very massive T cell activation. Okay, and the overproduction of the T H cell cytokines, causing the cytokine storm. So that creates a systemic toxicity. All right. So usually the immune response, which is the normal immune response, it is highly regulated, calibrated, and calculated. But in case of super antigens, they can create havoc inside the system. Now, superantigens can be exogenous superantigens, which are soluble microbial secretory proteins. Okay, they can be enterotoxins, they can be toxic shock syndrome toxins, and exfoliative dermatitis toxins. So, these are some of the examples of the exogenous soluble microbial secretory proteins. Now, enterotoxins by Staphylococcus aureus and Streptococcus pyogenes, they are very much, studied and very much founded here. So, these enterotoxins, they are thermostable, all right, and they can also withstand extreme pH. Okay, they are resistant to degradation by proteolytic enzymes, for example, pepsin and trypsin. So you can see, how much resistant these enterotoxins are. Moreover, they can retain activity even after cooking or the digestive process. Therefore, they can cause food poisoning, right? So even in your tummy, you have a very low pH because of the HCl, but they are resistant and therefore they can cause food poisoning. Even you are cooking, right? So usually say that cooking kills microbes. Yes, in normal condition. But when it comes to super antigens, then they are resistant, okay? And then they can survive and they can cause food poisoning. When we are saying survive, that means they can maintain their, the nature, the fold structure, all right? They are usually very small, 20 to 28 kilodaltons, and they are the two domain protein, right? And the sequence diversity is there if we compare Staphylococcus aureus and Streptococcus pyogenes. Then the sequence diversity is there 15 to 90%, but they are structurally similar from the both sources. So if you compare Staphylococcus aureus or Streptococcus pyogenes, they are structurally similar, although they have the sequence diversity. So you compare here, this is Staphylococcus aureus SEA protein, and this is Streptococcus pyogenes SPEC or SPEC protein. You can see, although they are different, they are two different sources, but still they are structurally similar. So you can see here, the helices and the beta sheets. Okay. Now coming to the endogenous kind of superantigens, so they are basically cell membrane proteins of the integrated viral genes. Okay. And one of the examples is the minor lymphocyte stimulating determinants, which we call them as MLs, and they are encoded by endogenous retroviruses, for example, the murine mammary tumor virus family. So this is one of the examples of the endogenous superantigen. So now, just to summarize for you, T cell superantigens are of two kinds, exogenous and endogenous. And exogenous are usually, secreted by Staphylococcus and Streptococcus species, while endogenous, they are the viral proteins which are expressed along with the host genes. All right. So that's the difference between the exogenous and endogenous. Now let us see what are the some effects of these superantigens. They can cause immature CD4 positive and CD8 positive T cells to become depleted. Immature cells can be depleted. They can cause that. Okay. But they can cause the proliferation of the mature CD4 positive and CD8 positive T cells. So immature cells are, they can cause depletion, but they can cause maturation of and proliferation of, the proliferation of the mature CD4 positive and CD8 positive T cells. And they can produce the cytokine storm, driving the mature T cells into the state of energy or unresponsiveness. All right. So when it happens, that gives the opportunity of other microbes also to flourish, because our T cells cannot respond. Right. Now, it can also cause systemic inflammatory response syndrome, which we call them as a sepsis. And sepsis is a very, very, emergency condition. All right. So it can create sepsis also inside the body. Another effect is the toxic shock syndrome, because you saw, in the earlier slide, that the superantigens can be of the exogenous origin, and they can create the food poisoning. So what is food poisoning? It is a toxic shock syndrome. It is a kind of toxic shock syndrome. So that is created by, or that is generated by, or that is caused by the exogenous kind of superantigen. Then scarlet fever, that can also cause scarlet fever, and atopic dermatitis, which you call it as a eczema. So now, the focus is to know whether the superantigens are, the reasons behind the atopic dermatitis or eczema, and so many studies have found that yes, they are the one of the reasons of the eczema. All right. So these were some of the effects of the superantigens. Now let us see these superantigens in therapy, how we can use in cancer therapy. So staphylococcal enterotoxin B, which is the SEB protein, it can strongly stimulate T cells. And when it can stimulate T cells, it may help the immune system to attack the tumors more effectively. So that can be utilized for cancer therapy. Then SEB, it has been shown to enhance the activity of the CAR T cells. All right. So CAR T cell therapies nowadays, it is one of the most advanced therapies for the cancer treatment. And that can improve anti-tumor responses when used together with the antibody-based therapies, or using the monoclonal antibodies for specific targeting of the tumors, right? So we can utilize these super antigens here, but it is not easy. There are major challenges, and the challenge is the toxicity. Okay, they create toxicity. Now they may cause dangerous immune overreactions because they are super antigens, so they can, and they do, right? So what is the strategy to avoid that? The first strategy is to split super antigens and divide super antigens into two inactive pieces. Then you attach two antibody fragments, and these pieces only reassemble into an active super antigen at the tumor site. So making the T cell activation more selective and avoiding such kind of overreactions or reducing the incidences of overreaction. That is first strategy. The second strategy is to engineer super antigen like proteins, okay, because we know the structure. So we can also create certain proteins and we can delete those domains which can cause overreactions, and we can utilize them for the cancer therapy. So these, they resemble structurally and functionally, but they produce less toxicity. So that is one of the ways to get, such proteins and utilize them for cancer therapy. All right, so that's how, this is how we use these super antigens in cancer therapy. So I hope, you enjoyed today's session. If not, I'm sorry for that. But if you have any question, you can just go through this video again, or you can use some text available, or you can write to me. All right, now I will meet you in the next class. Till then, take care and bye bye.